Hi, everyone. Thank you for joining today. We have three presenters, two faculty and one student. So I think, Dr. Hammond, are you going to start first?
I usually have a student presentation and then Dr. Jackson, I think.
All right, why don't we start with the student presentation? Are we all set?
Yeah, sure. Can people in the room hear me well?
Yeah, we can hear you.
Yeah, thank you. Let me share my screen.
I want to view the room as well.
Yeah. Can you see my slides?
Yes, we can see your slides.
Okay, I'll go ahead to get started. Hi, everyone. My name is Zhuolin, and today I'll present my thesis, a systematic review and meta-analysis on choroidal neovascularization in the fellow eyes of patients with neovascular age-related macular degeneration managed in the study eye. And my thesis advisor is Dr. Ian Seldenha. So I'll start with some background to walk you guys through it. And the first part is population. We are targeting patients who have age-related macular degeneration, also called AMD. And an important condition of AMD is having choroidal neovascularization. And then I'll talk about the treatment of AMD. So AMD is a chronic eye disease characterized by the slow deterioration of the macula, which is responsible for sharp and central vision. And it's the leading cause of irreversible blindness and is strongly associated with aging. And the recent systematic review shows approximately 200 million people are suffering from AMD. And with the global population aging, the prevalence of AMD will be anticipated to reach around 300 million by 2040. And in this slides, I will show you the progression from a normal eye to dry AMD and finally to wet AMD or neovascular AMD, which is the focus of our review. So here is the normal eye fundus photo, and the retina you can see here is free from drusen, which are yellow deposits, often considered as the early sign of AMD. So here is the dry AMD, which is the most common type, accounting for about 20 to 50, 20 to, no, 80 to 50, 80 to 85% of AMD cases. And it's marked by, like you can see here, accumulation of the drusen beneath the retina, while it typically can progress slowly, so it may only cause gradual vision loss. And it will progress to wet AMD, also called neovascular AMD. And this form is characterized by the abnormal growth of blood vessels known as choroidal neovascularization. And these vessels you can see here are prone to leaking, and it may cause scarring and more severe vision loss, and also hemorrhage in the fundus as well. And our systematic review will only focus on this type of AMD, neovascular AMD. And so how do we treat neovascular AMD? From the last century, physicians and scientists have been dedicated to investigating effective ways to treat neovascular AMD. So beginning of the 1970s, the laser or photocoagulation treatment is to seal the or destroy the abnormal blood vessels. And in the 1990s, submacular surgery and external beam radiation were explored as innovative approaches to manage the condition. And by the turn of the century, photodynamic therapy combined the laser technology with target drugs like waterporphine to improve outcomes. And the breakthrough came in 2006 with the first approval of anti-VEGF therapy, ranibizumab, and which transformed the treatment landscape by directly inhibiting the growth of the abnormal blood vessels and significantly improving the patient outcome. So these are all the treatment that we are looking at. And okay, let's move on to objectives. Our primary objective is to evaluate the incidence of CNV in the fellow eyes of patients with NAMD managed in the randomized control trials or well analysis non-randomized trials or prospective cohort studies. So why we only study fellow eyes in this review? So there are three main reasons to study fellow eyes. So first, NAMD is inherently a bilateral disease, meaning both eyes are at risk. And second, most of the clinical trials or cohort study often only focus on the study eye, so leaving limited data on the fellow eyes. Also, understanding fellow eyes progression and risk factors can improve monitoring, prevention, and treatment strategies for the patients. And the second objective is to investigate the potential risk factors associated with the incidence of CVNV. So here are risk factors that we are interested in, including the demographics, age, sex, and race, and the year that the clinical trials or cohort studies completed the enrollment, and the patient's best corrected visual acuity, BCVA, also the age, AMD-related morphology, and as well as hypertension. Okay, let's move on to methods. Here are criteria for selecting studies. Our study types are including randomized control trials, non-randomized control trials, and prospective cohort study. And for there's no participant size limitation for randomized control trial, but we do need more than 30 participants for each groups in non-randomized control trial as well as prospective cohort studies. And our patients include adults aged 18 and older with NAMD in the study eye. And importantly, their fellow eyes must not have evidence of CNV at baseline. And as I mentioned before, there are 5 types of interventions that will be included. And outcomes in the fellow eye include CNV incidence, treatment outcomes, and changes in best corrected visual acuity, BCVA. And here is my current progresses. We already conducted a comprehensive search across 5 databases and got over 20,000 studies. And after removing duplicates, around 15,000 studies remained. And at present, we have completed the title and abstract screening, and 1,276 studies have been identified for further analysis of the eligibility. And our next steps involve continuing the full text screening and extracting the data from the included study, as well as assessing the risk bias using validated tools. We will use ROBE2 for RCTs and Robin's I and E for non-randomized trial and cohort study, respectively. And after finishing the data collection, we'll measure the treatment effects. So for time to event studies, we'll use hazard ratio to assess the incidence of CNV in the fellow eye. For continuous outcomes like BCVA, we'll use mean difference or standardized mean difference, depending on the scale of the consistency. So for the dichotomous outcomes, such as vision loss, we will calculate the risk ratios. And the heterogeneity will be assessed in 3 domains. First will be the clinical heterogeneity, that include the variations in the patient's demographics and treatments. And methodological heterogeneity considers study design differences, while the statistical heterogeneity is quantified using the metric like I-square, I-square, tau square, and Q statistic. So if the I-square is higher than 50%, it will be considered as moderate to high heterogeneity. And the tau square will be quantifying variability across studies. And if the p-value is lower than 0.05 in Q statistic, it will indicate the chance-related heterogeneity. And our data synthesis involves 2 approaches. first narrative synthesis is to describe the patient's characteristics, methodologies, and outcomes. And second, if the meta-analysis is appropriate, we will aggregate the effect size across the studies, and we will use a random effect model to account for the heterogeneity and visualize results with a forest plot. And then we'll perform subgroup analysis to examine differences based on demographics, treatment types, CMV status, and BCVA in the fellow eyes, also the hypertension and AMD morphology. And sensitivity analysis will assess the robustness of our findings by excluding the high-risk studies and those with the missing data. And this will ensure our studies are reliable and valid. So this is all I have for today. And I'll appreciate if you have any suggestion.
Good job. Any questions or suggestions?
So I have, the first question is focused on hypertension as a risk factor. Is that because you believe you will have good access to that? When you haven't looked at other potential risk factors, systemic risk factors, or is it because you think it's most likely to be the one that will influence the risk?
I think that will be hypertension may influence the risk as we are studying the aging populations, and most of the aging populations, they might have the hypertension. So I just wondering if they are like related.
But most of the aging population has hyperlipidemia, and you're not looking at hyperlipidemia. I was just curious as to how you ended on hypertension as your one systemic risk factor. It is one that's likely to have data available within the study, so I think that's good. The second question around hypertension is, how are you going to define it? There are standard definitions of hypertension, but I'm not sure that these people were always measuring blood pressure. So are you going to define it as receiving an antihypertensive drug?
So first, the reason I choose hypertension is because if the patient have the hypertension, and also there's a fragile new growth blood vessels in the retina, and I think the hypertension may cause the leaking of this new blood vessels. So I think there might be association. And for the definition of the hypertension, I think if the results of the clinical trials or cohort studies, they report the patients have the hypertension, that's will be I'm looking at. So for the drug, I think it's a little bit hard to define whether those people have the drug.
Yeah, I guess this is a meta-analysis. So the issue may be if there's any variability in how it was ascertained or defined within the study. The second comment, and this is just a comment, is visual acuity is lost in AMD in two fashions. One of which is choroidal neovascularization, and the second of which is geographic atrophy.
Mm-hmm.
And you haven't really talked about geographic atrophy, and that's fine. You're looking at second eye choroidal neovascularization. But it's a potential confounder in the visual acuity analysis. Because the development of geographic atrophy in the contralateral eye could influence your visual acuity. And so it's not your primary outcome, but it may be useful to have that information included in your model for the visual acuity outcomes.
Yeah, I see. It's a good suggestion. Yeah, I'll include that in my analysis. Thank you.
I just have a quick question about some of the inclusion or exclusion criteria that might be specific to the trials that you might include. Are you looking at any of the inclusion or exclusion criteria that might be specific to the fellow eye? Because that sometimes comes into play with including participants in trials, especially for AMD. Other than free, I know you're saying that they're at risk, but no evidence of AMD at baseline, but there are no other criteria? Or are you checking to look at what all the criteria are for including or excluding participants with fellow in that setting? I'm just curious.
Yeah, that will be easier for the clinical trial when I screening the studies, because I can find their identification number and look at search on web and to see their inclusion criteria and exclusion criteria. Most of the clinical trial they will present whether the fellow eye have the neovascular AMD in their exclusion criteria. So for that part, I'm not that concerned, but for cohort studies, it's a little bit hard to decide whether the fellow eyes they include have the hypertension. So that's also my concern about that. So far I didn't find a best way to define whether the fellow eyes have the risk. I just reading through the paper, and if they say they have the evidence, I will exclude that paper. But if they don't say whether they have the baseline, I will include for now.
So you're still uncertain whether or not the fellow eyes are at risk, right? Based upon.
Yeah. Yeah, for the cohort study, it's still uncertain.
They're almost always at risk.
Yeah, but, yeah.
I mean, I don't know that you need to worry about that. The second eye is almost always at risk. The issue is not whether or not it's at risk, it's what's the anticipated risk is. And at least there are simple scoring systems for it, based upon the appearance of the eye for estimating the risk of the progression from intermediate stage to late stage AMD. Late stage AMD, which is what neovascular AMD is almost never de novo. It almost always progresses through the earlier stages. So these data are probably not going to be in your papers, and you're not going to be able to include them. But there are scoring systems where you can estimate the 5-year risk of neovascular AMD or late stage AMD, either neovascular or geographic atrophy, based upon the appearance of the intermediate stage AMD. So they're all at risk. It's just the question of what the magnitude of the risk is.
But I was also considering the fact that sometimes in AMD studies that I've worked on, we sometimes excluded participants based upon some characteristic of the fellow eye, even like visual acuity difference or something. I'm just vaguely remembering that from the past. So I was just wondering what heterogeneity there may be with the trials that are being included in this based upon the inclusion or exclusion criteria for the fellow eye. So you may have characteristically people who might be at inflated risk or not, depending upon the way they've structured the trials. That was my concern.
Yeah, that's a good point.
Any other questions?
Okay, I'll stop sharing.
Thank you so much.
Thank you.
Thank you.
Jeff.
Yeah, so Dr. Irvin asked me to give a very informal chalk talk about what I do. Most of you think not much. And so I'm going to start by talking a little bit about how actually my grants or the grants in which I participate are funded. I've been doing clinical trials since the late 1980s. And I think on every single clinical trial I have done, save one, Janet Holbrook has been involved. So we have worked together for a couple years now. Almost all of the trials which I have done, I think I'll pick all, have been NIH funded. And the NIH funding mechanism is usually a UG, it used to be U10s, now it's UG1s. And when they come out of the National Eye Institute, they come with at least 2 and typically 3 linked applications. One is the chairman's office. Chairman, and these grants are 100+ pages. They're really so much fun to do. The chairman's office funds myself, usually some supporting staff, and sometimes some other clinicians that may be involved in the management and running of the trial in a way, and I'll sort of give you some examples. The second grant is the coordinating center grant, and currently the trials I'm working on, Elizabeth Sugar and Janet Holbrook are co-directors of the coordinating center. For the National Eye Institute, the coordinating center encompasses both the clinic coordinating center. That's the group that manages the individual clinical centers and handles all the contracting and does all of those sorts of things. And the data coordinating center, which does the data analysis. Not every institute does it that way. I'm actually a co-investigator on a grant that's in development, a project that's in development. It's funded, but it's in development right now out of NIAID, in which the CCC, Clinic Coordinating Center, and the DCC, the Data Coordinating Center, are independent contract research organizations. And we may think that unusual because we always merge them. That's what we do. Although, Anne, have you been part of a DCC without being part of the CCC?
Yeah.
No, you've never had. Yeah. I don't know. I think you may have with the
COVID studies.
yeah, the COVID studies. And so we don't like it because they're very much linked, but there are places that do that, and NIAID typically does that. The study chair
Is
has,
that NHLBI too?
NHLBI does as well. The study chair has several good study chairs have several roles. I have seen study chairs who are figureheads, okay, where a lot is done and they tell you what they're told to tell when the slides are handed to them at the meetings. But a good study chair has, first and foremost, formulates the question. For industry-funded trials, which are phase 3, the question is usually very simple. Does it work? Right? That's what they're asking. Does it work? And can I then market it? Right? And so, but phase 4 or comparative effectiveness trials, which is most of what I do. I have done phase 3 trials, but most of what I do is usually, does it work better? Right? And I tend to think of these things very simplistically, but it enables you to sort of cut through a lot of what you think about more formally to understand, what we're asking. But does it work in diseases where there are treatments becomes a very tricky question in a phase 3 trial. Often, we can only allow a short time of placebo observation or sham, which is the 3 ways which you might defer therapy. For many of the cytomegalovirus retinitis trials we did, we deferred therapy and located and excluded patients who had immediate vision-threatening lesions when we wanted to see if it worked. And the deferral was usually for a month, no more. And then we would start therapy. But the disease was so rapidly progressive, we got answers in that time frame. And then we would follow longer for side effects and things like that. I am still a practicing clinician. I see patients a day a week, half a day a week. That does 2 things. One, it helps assist me in formulating questions, right? I often, when we do a trial or when we do a study, it's what do I want to know to manage patients better? That's sort of my starting point. The second is it keeps me abreast of sort of the nuances that become very difficult when you have to codify them in a protocol, in a data collection form, in all of those things, because those decisions become arbitrary. And you'll find, as you work with clinicians don't like being arbitrary. And by and large, they think, well, what about this? What about, and they think about the individual patient and the exclusion, and you can't do that. At the end of the day, you have to have an outcome that's arbitrary, progression more than X microns. Well, what if it's X plus 1 micron, And the answer is, well, that's too bad, we got to draw a line somewhere, and so it's also thinking about what's meaningful and what is arbitrary. So the first thing that I think a good chair does is formulate the question, and then also assist in how you're going to address that question, right? There's a study design that's now used in uveitis trials, which is what I typically do now, in which patients are placed on high-dose oral corticosteroids, which are typically the initial therapy, and then rapidly tapered to 0 over a time frame that's not what's done in clinical practice. And the second agent to evaluate their efficacy is started also at the time of the corticosteroids, and then it's a time to relapse study, right? And the second agent prolongs the time to relapse, and that tells you it works, right? But does it tell you how to use it? Does it tell you does it work better than the standard therapy? Does it tell you anything about those? No. And in fact, the current trial that we're doing, the ADVISE trial, came exactly out of that. That rapid taper to 0, they did 2 studies, showed that adalimumab, the most lucrative drug in the history of the United States, possibly of the world, was effective for the treatment of non-infectious intermediate posterior and panuveitis. You don't need to worry about those. And we said, okay, it works, but should we use it? Does it work better? And so the ADVISE trial is adalimumab versus conventional immunosuppressive agents for treatment. And it's really designed as a comparative effectiveness trial, with every effort for the management to be exactly what we would do in real life. And yes, we make certain things more rigid than some clinicians would like, but that's what it is. So, and that I think is important. I actually bristle when people get up and say trials are artificial. And I bristle because, in fact, many of the comparative effectiveness trials that we've done are pretty close to what I do in clinical practice. And so I don't think the trials have to be artificial if a comparative effectiveness trial is well designed. And you shouldn't automatically accept the party line that they're highly artificial. Yes, there is selection bias because not everybody participates in all those things. But the best comparative effectiveness trials try to emulate what people do in the real world. The third place where I think good chairs, and I say this because a lot of chairs don't want to do this. When you define outcomes, right? As I said, it's arbitrary. What are you going to call, when we used to do CMV retinitis trial, what are you going to call progression? How much are you going to allow that retina to get chewed up before you say that's a meaningful difference? What are you going to call worse something? What are you going to call reactivation if you're doing a time? What are you going to call this? So you can make those definitions often based upon literature and what people have done before. And a lot of times industry uses what's been done before. And they do that because, at least in my experience, the FDA values very strongly internal consistency.
Yes.
Because then they are treating every company the same. And that's an important principle for them. And at least that's my observation.
It definitely, it's the only place where I'm encouraged to copy and paste.
Right.
So that way things are congruent between different places.
So the downside of doing what's been done before is sometimes you perpetuate a bad idea. And I'm not saying that the FDA does that, but I'm saying that's the risk. Or times change. And so for example, for a long time, we were willing to say 10 milligrams of prednisone a day was an acceptable level to get to. We now say 7.5 because data have come down the pipe that you have to be at 7.5 or below. have effective minimization of long-term steroid side effects. So sometimes data evolve and you change what you want to do because it's evolved. So a good chair, in my opinion, helps with the formulation of outcomes. And I actually work sometimes with the coordinating center on forms, one of my least favorite activities, but it's important to think about how are you going to question, how are you going to assess? And under optimal situations, if you have a coordinating center that's been doing the job for a long time in that field, and a chair who's been involved in trials for long, it becomes a very smooth conversation. New chairs have a tougher time because they're just not used to some of the stuff that you have, the sort of arbitrariness of how you're going to define it, and they really think about the clinical nuances. And that is one of the differences, of course, is the clinical nuances. And then lastly, I think obviously when you interpret the data and you have to place it in context, a good chair is critical for both the introduction and the discussion of any manuscript. We're writing the advised results up now. We finished follow-up, we're doing data cleaning, we're writing up the results. And there are data since the design of this trial. The trial went on for 6 years. Well, this is the 6th year. Since the design about adalimumab that make us worry about a simple conclusion. Namely, if you give people adalimumab for a real long time, they start getting antibodies because even though it's a fully human monoclonal antibody, it's still not their own antibodies. And so those antibodies reduce effectiveness of the drug. And there's some data that combination therapy might be better. So we worry in a study in which 1-year follow-up is provided, are we going to say something that might not be true if we had 5 years of follow-up? So I think a good chair is able to integrate outside sources of data to make sure your interpretation of the results is meaningful and hopefully not erroneous, but you know what I mean? It's you've nuanced it appropriately. And so I think that's the other place where a good chair really can contribute to this. You are going to learn a lot about being a CCC and a DCC. And the mechanics of running a trial is a science in and of itself. I mean, it's sort of almost an implementation science, right? The mechanics of running a trial. And I would argue that the people I've been fortunate to work here are among the best in the world. Really, this is the place to train, to learn how to do those kinds of things. And data analysis, again, outstanding data analysts. Although I will say, I've been doing this for so long that I've seen the principles of data analysis evolve and become more statistically rigid. Probably better, but they evolve over time. Have you seen the evolution?
I would agree.
Yeah. You're a little younger than am I, but
Yeah.
so that's half as long as I've been.
Oh, there you go.
But yeah, but so and you'll obviously have to keep abreast and so forth. But I think that's the sort of the roles which I see myself play as a study chair for most of these. And I'm happy to entertain any questions.
Thank you. Oh, that was great.
Any questions?
I have a question, purely curiosity.
Yeah.
For the drug sponsor, it sounds like your trials are all NIH funded.
Yes.
So then do the drug sponsors like agree to provide the drug or how's that?
So we do.
Because I
Yes.
could imagine they would be a little nervous about a head-to-head trial with a competitor.
So we have been again fortunate over the past 20 years of doing it. Actually, ever since I started having industry of the test drug supply it, and sometimes it's one of the comparators. However, so for example, in Advise, AbbVie provided the adalimumab. NIH was never going to pay for it, and for any of it. It did not provide, nobody provided methotrexate, prednisone, mycophenolate. These are the conventional agents that would be the comparators. But methotrexate, comparatively speaking, is cheap. It's in the hundreds of dollars a year, or prednisone is hundreds of dollars a year. Methotrexate is somewhat more than that, but not a lot. Mycophenolate, which used to be expensive, is now a couple thousand a year. By comparison, adalimumab recently upped the price to 80,000 a year. It's the most prescribed drug ever because it's so effective.
Does the insurance play?
Yes, in clinical care. What we have done and what NIH has allowed us to do is to create a small emergency fund to assist the individual clinical centers in assisting with patients with expenses that would otherwise keep them out of control.
Like parking.
Like, well, yes, there are two things. One of which is there's usually a small visit fee that we give to each center to pay for parking and childcare and transportation and all those things so that the patients can come. But there's also a second one. If the patient gets overburdened with copay, there are ways to help them with that. And so we tried hard to make it easier for patients to participate. Now, I'm not sure that every institute does that. But NIH policy, for those who don't know, is that they do not pay for those expenses that would be normally paid for as part of clinical care. They pay for only those expenses that are incurred as a consequence of participation in the trial. And so to the extent that you can take expensive things off plate for the patient, you encourage participation and you improve generalizability, right? If you have people with crappy insurance with large copays, and those are going to be often socioeconomically less well off portions of the population, then you decrease your generalizability. So that's the other thing. You do want to, particularly in a comparative effectiveness trial, you want to be as generalizable as possible.
Does that usually involve like randomization, or it's based on clinical judgment?
Well,
The trial
they're all randomized.
or randomized.
Yeah.
Okay.
When people ask me what's the best way to treat a patient, I always say randomly. I have done cohort studies, and I've done some long-term cohort studies, but most of all my trials have been randomized.
Okay.
Sorry?
I have one more question for you, but I'll ask it some other time.
Well, we're setting up.
Oh, yeah.
Yeah.
So I hope to, I think at least a lot of students that's coming later than me ask me what they can do if they want an RA position, or like if they want to participate in your study, what they usually can do.
So the chairman's office grant is usually a very small grant. So there's no real funding for a student, and the chairman's office really doesn't handle the mechanics, nor does it teach you the things that you're here to learn. You really should be reaching out to coordinating center directors. And if you want to work, Advise is now in the final year, but if you had wanted to work on Advise, you would have reached out to Janet Holbrook and or Elizabeth Sugar, and then you could have perhaps been integrated into the trial there. And so when anybody comes to me and say, we'd like to work, I always refer them to the coordinating center director, because that's where you will learn the stuff that you want to learn.
But I think it's also interesting, if you have a focus on a specific clinical area, it never hurts to be able to have some of that in-clinic experience and learn a little bit more about what all the types of measurements, because that's where I learned so much about ophthalmology, is actually working in the clinic, not working hands-on. I learned like how to do all the procedures, learned how to measure visual acuity.
Yeah.
I followed the clinicians in clinic, I worked in clinic, and that kind of helped me as a clinical trialist, because then I understood all the clinical stuff. And I could be able to, you could sit at the table with the clinicians and at least understand what they're talking about. So that's.
I couldn't agree more with that statement. The best clinical trial coordinating center people are those people who understand the subject matter. And Ruth Ann and I have an affiliation with the Dana Center over in the Department of Ophthalmology, which tends to be more epidemiologically focused than clinical trials focused these days. But there was a time when they were doing trials of treatment of trachoma and or onchocerciasis, typically overseas. And because they were doing trial after trial, even the people who were non-clinicians became very adept at understanding the clinical questions, and really was very easy for the clinician and the non-clinician to talk in that situation. I would argue that Sheila West can interpret a photo of trachoma far better than any clinician I know, just because she did it for so long. And so that is true. I fully agree with Anne that the more you know or understand about the subject matter, the easier it is to have a conversation with a clinician.
I think there was one comment in the chat. Oh, that popped up.
Like gastroparesis and MASH groups could be places for student work. These groups are large network structures from multiple simultaneous studies. They're doing trials right now, but have in the past and probably will in the future.
That makes sense. Thanks. Thanks, Dave. That's good to know.
Well, actually, speaking of MASH, I'll get started with my presentation. So it's good. For those, I think most of you know me, but my name's Zhuolin. I'm a physician and a medical officer at the Food and Drug Administration, and I also come here on Thursday afternoon to be a guest instructor here. So I'm going to talk a bit about what the FDA does, and just go through like a potpourri of different topics related to it. Okay, so we'll start out with what the mission of the FDA is, investigational new drugs and how we're involved there, evidentiary standards for approval, type of endpoint measures, Dr. Jones was just talking about, they can be arbitrary or non-arbitrary, and then how to look up public information related to the FDA, including labels and advisory committee meetings. So the FDA is really a public health organization, and it works to assure the safety, efficacy, and security of human and veterinary drugs, the food supply, cosmetics, and other products that emit radiation. And really what the FDA is trying to do is advance public health by helping to speed innovations and make them more effective, safer, and affordable, even though we don't necessarily work with drug prices, so don't put that on us. So the impact of the FDA's mission is huge. About 1/5 of every dollar in the US GDP is under the regulatory authority of the FDA. As an FDA employee, I see most of the information I work with is confidential information from drug sponsors. So information not in the public domain. And as a federal employee, I'm expected to uphold the law at all times through all means of communication. So let's go into kind of the nitty-gritty about where a drug's relationship with the FDA begins. And this is where we're going to talk about INDs, which are investigational new drugs. So here's a timeline of drug development. I'm sure everyone here is familiar with that. So once basic research and preclinical development have been completed, that's really where the IND comes in. And that's when a drug sponsor, either a pharmaceutical company or sometimes an academic investigator submits an IND. And this is really where the first interactions with the FDA come into play. And the FDA reviews an IND, so that's basically we call them sponsors, plan to test the drug in clinical development, so phase 1, 2, and 3. And we're really looking to make sure that the rights of human subjects are protected and to ensure that the quality of the clinical trial is adequate to evaluate the drug's effectiveness and safety. So I'm just going to call it an IND, but the abbreviation is there. So it applies to all drugs and it needs to be submitted to the FDA if it's not previously marketed, if it's not previously authorized for marketing in the US and it's intended to be used for the purposes of clinical investigation, or for the purpose of clinical treatment when no approved therapies are available. And you can read there what exactly the definition of a clinical investigation is, but I think we generally have a good sense of that. So what I do think is more interesting is what defines a drug. And here the most key point is the intended use of the therapy. So a drug is something that is intended for use in diagnosis, cure, mitigation, or prevention of disease, or anything other than food that's intended to affect the structure or function of the body. So here's an example. Essential oils, we all see them in the Whole Foods sections. They're popular, they're widespread, they're used. Are they a drug? So if you're just using them to kind of make your house smell better, that sort of thing, it's not technically under the definition of the drug. However, if you start marketing it with saying, oh, this can help you quit smoking, or this can help you sleep, then you're trying to either mitigate or treat like tobacco use disorder, a disease. So really intended use is extremely important for the FDA. So INDs really live, the reason that they exist is because the FDA has authority on when a drug is shipped across state lines. Even if your drug is made and going to be distributed within a state, you still need an IND, but that's where this full law came from. And so, yeah, this is the exception. An IND is how you get an exemption from the FDA to ship an unapproved drug across state lines. Kind of covered this, but really the reason to have an IND is just to make sure that human subjects are being safe. There's safety in these trials, especially in first-in-human studies. And then as you progress through clinical trials, making sure that the safety and efficacy is being well studied. I just want to emphasize here that INDs are only one layer of protection. IRBs and informed consent also help protect patients, and those are required for any clinical trials looked at by the FDA as well. Here's kind of where the authority or where all our recommendations come from as a regulatory agency. So there's laws that are passed by Congress, such as the Food, Drug, and Cosmetic Act. Those are written into regulations, so the Code of Federal Regulations that's divided into 50 titles. Title 21 is Food and Drugs, and for example, Section 312 pertains to INDs. And then from the regulations, the agency writes guidances. And I think guidances are really important, and these are publicly searchable. So here's an example of two. So if you ever have something related to drug development that you're interested, like what is the FDA's viewpoint on development of some of the drugs for disease for like an ophthalmologic therapy? Chances are there might be a guidance on it. So I work in the Division of Hepatology and Nutrition. We have several guidances around what we expect endpoints to look like, trials to look like in liver diseases. So again, these are all publicly searchable. You can just Google FDA guidances, click the first link, and then type in what you might be curious about. And it'll show you even things related to your search. So even if you don't have the exact search terms, these are really helpful. So next, I mentioned one part of an IND is to have studies that are safe and effective, but what is the FDA really looking for in terms of efficacy? So this came up a few weeks ago when we had the journal club for Alzheimer's disease. Someone asked, why were there 2 adequate and well-controlled trials versus just 1? And as Dr. Jobs mentioned during that time, that is the law. So the law says that we require 2 adequate and well-controlled trials to distinguish the effect of a drug from other influences, such as placebo effect. However, in 1997, the FDA Modernization Act was passed, and this can allow for 1 adequate and well-controlled investigation and confirmatory evidence. What confirmatory evidence is depends on each situation, and that comes from conversations with the FDA. So it can be mechanistic or pharmacodynamic evidence, clinical evidence from a related condition or population, relevant animal model, etc. I'll say I've seen a lot of different forms of what confirmatory evidence looks like, but these are conversations we have with the sponsor well before kind of that determining of effectiveness for what confirmatory evidence might look like. So, and that's where regulatory flexibility comes in, where we're allowed to exercise our scientific judgment in each individual development program. But we try and be as uniform as possible, especially amongst the same disease space. So adequate and well-controlled trials. One thing I learned pretty much coming from the FDA is while some observational trials are used for some things, the FDA really wants placebo-controlled, randomized controlled trials. So that's kind of our gold standard, and other things are sometimes used maybe when that's not possible. So here I just kind of list that out there. In the interest of time, go into it. So the other thing I think is really interesting is endpoint definitions. So as Dr. Jobs was mentioning, sometimes these can feel a little bit arbitrary because you might be cutting like a disease off at a certain time point. Like for blood pressure, is there really a difference between 141/82 versus 140/80? Probably not that stark of a difference, but you have to draw a line somewhere. What's most important about an endpoint is that it affects the way someone feels, functions, or survives. So if there's an endpoint that's not directly affecting the way someone feels, functions, or survives, it's determined to be a different kind of endpoint, and we'll go into that. So one example of an endpoint is clinician-reported endpoint measures. So this might be like clinician observation and interpretation. So here, like for example, if you're doing like a derm drug, it might be how small did the rash get within that phase of therapy. There's also patient-reported outcome measures, and these can be highly variable. So these can be symptom-related. That goes into the feels of the feel, function, survive. So if someone has irritable bowel syndrome, IBS, one outcome could be a patient keeping a diary of their symptoms and do they feel better with the drug use or not. There's also biomarker endpoints, and these are the ones where I was kind of alluding to where it might not necessarily affect how someone feels, functions, or survives. So for example, blood pressure is one. People with high blood pressure, they tell me all the time, in clinic, they'll tell you all the time, I feel fine, doc. I don't need anything. And it's not impacting how I function. But we do know now through, having good information about hypertension that over the long term, it does affect how someone survives. And so that makes it a surrogate endpoint. So it's on the pathway of one of these feel, function, survive. So blood pressure would be an example of a validated surrogate endpoint. We know for sure that affects the outcome of survival. There's also reasonably likely surrogate endpoints and intermediate clinical endpoints. So I'll get into some, there are some examples here. So in the liver world, we actually have a lot of reasonable likely surrogate endpoints. So primary biliary cholangitis is one where ALP, it's a liver biomarker, is used for reasonably likely surrogate endpoint. And in NASH, we use a biopsy. So sponsors can conduct trials that land on a reasonably likely surrogate endpoint, and they can get what's called accelerated approval for this, and I'll go into what that means. But in order to get full marketing approval, it had to have been on a validated surrogate endpoint. So I'll go into really quickly these expedited programs, including covering accelerated approval. Expedited programs are available if it's a serious condition, the therapy might provide a meaningful advantage and there's an unmet medical need. So accelerated approval, again, hits on a reasonably likely surrogate endpoint. So for example, in NASH, if there's improvement in NASH on biopsy, a sponsor can obtain accelerated approval for that. But as part of the approval, they must complete the clinical outcomes trial. So that is looking at death from liver causes and different clinical outcomes. Those can take a lot longer to form in some diseases. So this is a way to get a drug to market and get therapy to people a little bit sooner. So last thing I'll say is there's